For that introduction, Leanne. Um, and I hope that you can all see my slides and my laser pointer. So yeah, like Leanne shared, I will be presenting some of the work that I've done in my PhD, mainly focusing, I guess on this paper that Alex must have presented at ANS in Perth in 2024. And so this will be largely, will form the large part of my talk. But like Leanne also said, I did recently submit my PhD, so I thought it would be quite interesting to talk about some of the follow-up studies, thank you, that I've done from that. So like I said, I'm part of the Healthy Brain Aging and Plasticity Lab at UW and Perron Institute in Perth. And so one thing I'm interested in is looking at the effects of age as well on TMS. So I've got work about that and as well translating that into seeing whether this occurs in the human brain. So I've got some exciting stuff as part of this talk. But first of all, I'll just do a little quick introduction as part of this kind of talk into my paper. I'm sure you all know what rTMS is, but basically repetitive transcranial magnetic stimulation is a form of non-invasive brain stimulation that is able to generate these magnetic fields through the induction of a current through a coil, and these induce a secondary electrical current in the brain. And so the repetitive delivery of these pulses is what actually enables this method to modulate neuroplasticity in the brain. And so we know that rTMS is used widely in the treatment and the study of neurological disorders, and trying to reduce neurological decline as well in the case of healthy brain as a tool to promote healthy brain aging. But one of the things is that there is very limited underlying knowledge of the cellular and molecular mechanisms underlying these therapeutic effects. And so a lot of my work is focused on trying to understand the neuroplasticity mechanisms that are occurring inside the brain. And so as part of that, a lot of my work is preclinical and kind of focusing on that cellular level. As well with rTMS, we know that there is a lot of variability in kind of the presentation of how people respond to stimulation. And that can also be because there is so many different rTMS protocols available. You've got your simple patterned and complex patterned rTMS, each thought to induce different effects on neuronal excitability as well, albeit with some substantial variability. And as well, the brains is incredibly complex. So we have heaps of different brain regions involved in different kind of neural circuits and pathways in a lot of these disorders. And even within the cortex, you have different cortical layering, and within each of these layers, a lot of different cell types that are involved in producing this overall effect that we see with rTMS. So with that, understanding how different brain regions respond to different rTMS protocols is incredibly important, because things with like the motor cortex and sensory cortex has all these cortical layers with different functions. And so how iTBS or cTBS responds to or affects cells within these brain regions is something that I really wanted to try and unpack and understand with this paper. And so in order to do that, I come from a genetics background. And so I wanted to use the tools or the study of transcriptomics to kind of really understand what is occurring in these cells following rTMS. And so transcriptomics is the study of the expression of genes or RNA transcripts at a specific point in time. And so changes in these expression of RNA can tell us a lot about what is happening to dysfunction of cells following treatment, so in this case, stimulation. And largely this can be done through what we call bulk RNA sequencing, where it gets an average, where you have to homogenize tissue, and then from that, you get an average expression of multiple cells in a tissue sample. And so that can tell us on kind of a gross or mass scale what the effects are following, say, in my case, stimulation. But as well, one thing that I've already mentioned is the brain is extremely complex. So while bulk RNA could give us maybe a global outlook or a look of the global scale of how rTMS is changing a region in the brain, understanding and picking apart what is happening on these different cortical layers is also incredibly important, because we do know that rTMS does have cortical layer specific effects as well from previous studies. And so one tool that recently came out in 2016 and has slowly gained momentum within the field of neuroscience research, especially in particular, is spatial transcriptomics. And so what this is this tool allows us to combine this kind of bulk RNA next generation sequencing with histology to study the expression of genes in tissue and be able to map out exactly where these gene changes are occurring. And so this is an incredibly powerful tool and kind of recently got commercialized by this company called 10x Genomics. And so this is a lot of what I work with. And so this is an example of what the spatial transcriptomic slide looks like. It looks quite simple and plain and looks like a normal glass slide, but within each of these capture areas, there are almost 5,000 capture spots, each containing uniquely barcoded what we call oligonucleotides. And these are able to capture RNA released from the tissue exactly where they're released. And because they're barcoded in the analysis process, I'm able to map out exactly what gene expression profiles are occurring within each spot. And so with that, combining sequencing and normal H&E histology image also on the slide, I'm able to then generate what spatial transcriptomics kind of looks like, a result like this, where I can have spatially resolved different gene expression profiles across the brain. And you can see it really nicely can distinguish between a lot of the major brain regions that we see in the mouse brain. And so as part of this paper, I kind of had two objectives. First, to see how different rTMS protocols affect gene expression in healthy young adult brains. And as well to see how rTMS induces gene expression and how this differs between different brain regions in the mouse brain. And so we use normal black male mice as part of the study with rodent-specific coils that were designed by my supervisor, Dr. Alex Tang. And we've used previously in our labs to show plasticity changes in the mouse brain on both a cellular and behavioral scale as well. And so with these rodent-specific coils, one important thing to note as well that is mentioned as part of the title of kind of this paper is that we are delivering sub-threshold intensity stimulation. And so what that means, or how we define it, is just stimulation intensities that are below the action potential firing thresholds of neurons. And so with this stimulation, I wanted to look at two kind of main rTMS protocols that are used clinically, these cTBS and iTBS stimulation protocols. And so I performed bulk RNA sequencing to measure the global gene expression changes following these two stimulation protocols. And with the same stimulation protocols as well, use spatial transcriptomics to kind of get this mapping of brain region specific gene expression changes. And so kind of to run you briefly through what my experimental time might look like, it involved kind of 7 days of habituation for our animals and then 7 days of handling to make sure that they got used to the stimulation coils. And we decided to deliver a single session of iTBS and cTBS to these mice. And 3 hours following that, we performed bulk RNA sequencing alongside spatial transcriptomics as well. So these were two different cohorts of mice that we used. And we picked a 3-hour time point because it aligned with a lot of the previous kind of cellular studies that we've done, seeing plasticity changes following iTBS and cTBS. And so following sequencing analysis, I'll kind of run you through what of the results I found. So my bulk RNA sequencing results of I kind of focused on looking at the sensory motor cortex as well as a region for bulk RNA sequencing. And so looking at that region following iTBS and cTBS, we actually saw quite a diverse, kind of no clustering or no distinct clustering amongst my samples, which kind of indicated to me there is a lot of innate heterogeneity amongst my mice. And despite that, I still wanted to look at what performing differential expression analysis would kind of review. And looking at these kind of volcano plots, we use these a lot in transcriptomics. And this tells us the log flow change at the bottom tells us if there's an up or down regulation in the gene. So whether the expression of that gene is increased or decreased following stimulation. And this on this y-axis, it tells us the kind of the p-adjusted value of that change, so how great it was. And so, as you can tell from my volcano plots, we actually found no change in expression of genes following iTBS, which kind of matched my PCA plot, seeing that all of my samples were quite, didn't cluster well together. And following cTBS, my volcano plot actually found a predominant downward regulation of genes. And a lot of these were in kind of different DNA and signaling regulatory genes. So no evidence of kind of plasticity changes, which I guess was surprising. But from this, we kind of went with two kind of hypotheses. One, either there was no effect of, or no kind of big gross effect of stimulation, either both iTBS and cTBS, following at the specific time point we looked at. But it could also possibly, another hypothesis we had was that maybe the gene changes were more subtle than what bulk RNA sequencing could pick up. So like I said, bulk RNA sequencing takes the whole kind of cortex and homogenizes it and gets an average expression with gene changes. So we hypothesized that maybe the gene expression changes in maybe plasticity kind of pathways that we were kind of interested in wasn't actually occurring on such a global scale that you can pick it up in bulk RNA sequencing. And so this is where my spatial transcriptomics results come in handy and are quite interesting. And so this is an example of what kind of my brain section would look like on one of our spatial transcriptomic slides. And so looking at the same time point, just in a different set of animals, treated with iTBS and cTBS, you can see that across different groups, we have quite similar gene expression profiles and cortical regions that are picked up. And so what this then means across all my samples is that it provides me confidence to be able to perform differential expression analysis because there are similarities, similar number of spots or gene expression profile in each kind of distinct cortical region that I want to look at. And so really nicely, we were able to pick out different cortical layers and subcortical structures in the region of the brain that I looked at. And I ensured that they were all kind of from this similar plane as well. And so performing differential expression analysis from each brain region, you can see that we actually saw a larger number of differentially expressed genes following cTBS compared to I, which I guess kind of lined up with my bulk RNA sequencing results, where I also saw no gene expression following iTBS, but some downregulation following cTBS. And so one thing that was also quite interesting was that we actually saw sometimes similar or greater number of differentially expressed genes in subcortical regions compared to cortical. And so within these, there were changes in a lot of cellular regulatory functions, mitochondrial functions, but as well some synaptic and non-synaptic plasticity mechanisms and myelination, especially in the white matter tracts. So the data from this is obviously quite big, and so I haven't got time to kind of present it all, but a lot of it is in the supplementary material in the paper if you want to have a go or have a look at the specific genes kind of altered following these stimulation protocols in the subcortical region. But because our stimulation targeted, I guess, the cortical region or the cortex would have received the highest intensity of stimulation, we were interested in seeing what the specific nuances of stimulation effects were in the different cortical regions. And so with the mouse brain, the anatomical positions of these cortical regions are really well established. So one thing that I did with my spatial transcriptomics results was aligned it with the Allen Brain reference atlas. And this then allowed me to subset specific motor and somatosensory cortical regions as well. And now going forward, this then allowed me to pick out specific cortical region effects of stimulation. And so as you can see, this is just a graph indicating the different kind of number of spots or regions that align with the motor and somatosensory cortex following this alignment of each sample. And so this allowed me to perform differential expression analysis within each, comparing the cTBS with the sham or iTBS with sham. And so from that, you can see that there are distinct effects of each TMS protocol within each cortical region, but there are, and some of them overlap, but there are also distinct cortical region specific effects as well. Again, looking just purely at the numbers of differentially expressed genes, we see that cTBS in fact had a greater expression on gene expression profiles across both motor and somatosensory cortexes compared to iTBS. And so I'll go through some of the specific cortical region gene changes that I found as well. And so one thing that I can do with spatial transcriptomics is I'm able to map out, I guess, the levels of expression of specific genes across the region of the brain that I'm looking at. And so I did this with some of the notable genes that we found to be altered within each of the stimulation protocols in the motor and somatosensory cortex. And so this is one of the figures that I have from my paper. And so, as you can see, in the young adult brain, following iTBS predominantly, only following iTBS, we saw a downregulation in kind of these MBP, and MBOP, which are kind of notable myelin-related genes, following iTBS in only the motor cortex. And then as well, we saw downregulation of genes involved in synaptic intrinsic mechanisms. And so some of these gene changes I've displayed here, just involved in things like neurotransmitter release or synaptic function, and different voltage-gated ion channels as well, following cTBS, both in the motor and sensory cortex, but not following I. And so as you can see, there are differential mechanisms involved in each of these stimulation protocols in specific brain regions as well. As well, one of the things that I was able to pick out with my spatial transcriptomics results is that following cTBS, we did also observe alterations in notable excitatory and inhibitory neuronal markers. And so, as you can see from kind of my spatial plots, cTBS induced a decrease in expression of CAMK, which is a marker for excitatory neurons. And there was actually a differential regulation of inhibitory neuron subtypes in the same cortical region following cTBS. And so what this could mean is that there are multiple different mechanisms at play following these different specific rTMS protocols. And so trying to understand these maybe on a greater level, say at a protein level and a functional level, would be quite interesting moving on from this work. As well, with spatial transcriptomics, I could then pick out the specific cortical layer effects following each stimulation protocol within these cortical regions. And so how I did that was, because I've already isolated the motor cortex region for each sample, I could further subcluster that through unsupervised clustering, and it could actually pick out the different cortical layers, as you can see in this image. And so from this, again, I was able to perform differential expression analysis, finding, and within these specific genes altered, what I did as part of kind of trying to see what different neuronal processes were altered is to go through each of my gene lists and look at whether there was whether it was involved in a relevant neuronal plasticity function as well. And so what I did was categorize each of my gene lists within each cortical layer. And so from this, as you can see, within kind of the motor cortex region, each cortical layer has a specific set of genes that are altered following stimulation at the specific time point. And so just at a gross level, cTBS is because it's also able to alter a wider number of differentially expressed genes. Within these genes, there's also a lot of different processes, plasticity processes that are altered following cTBS as compared to iTBS, which could be maybe more specific in its effects. So things like synaptic plasticity and myelin was predominantly altered in iTBS, kind of these main genes that I saw. As well, within the somatosensory cortex, I did the same thing and looking at the number of differentially expressed genes overlapping each cortical layer within iTBS and cTBS. Again, we see that cTBS is able to alter a wider range of diverse plasticity mechanisms, whereas iTBS's effects are maybe more specific to synaptic plasticity or myelin plasticity as well. And so with kind of the bulk of knowledge or data that I have from this paper, what I showed was that, in fact, bulk RNA sequencing, because while it displayed little to no effect on gene expression within the sensory motor cortex, through spatial transcriptomics, we were able to detect more subtle, nuanced interaction effects of iTBS and cTBS that are brain region and cortical layer specific, and as well as specific to different neuronal and non-neuronal plasticity mechanisms. And so from kind of the spatial transcriptomics paper that we've published, we hope that it provides a really good resource to further study a lot of these kind of different plasticity mechanisms that are occurring in specific regions of the brain. With spatial transcriptomics being looking at just RNA transcripts at a specific time point, a lot of these transcripts could change if you looked at a later time point, or with different rTMS protocols as well. And so whilst this isn't definite, well, this paper doesn't really show a definite functional relevant outcome of simulation, it does give us a good platform to go and further investigate using things like patch clamp to see how these stimulation protocols are affecting specific regions of the brain now. And so from this paper, like I mentioned, one of the things that I kind of did in follow-up to that was to look at the effect of age. And one of the things that has been an interest of mine in my PhD is how aging does affect influence the effects of rTMS. And so as we know, there are several neurological changes that occur in the brain with age. Not only are there structural differences as well, but we do know that there is actually a reduced capacity for neuroplasticity in an aged brain. And this has been shown in the field of rTMS, where rTMS has been shown to be less effective at inducing neuroplasticity in older compared to younger adults. And so one of the kind of follow-ups I did from this was to see how aging affects these neuroplasticity mechanisms that I saw in kind of my young adult cohort, but in an aged brain. And so me and Alex then published this paper at the end of last year as a follow-up to the initial paper that I talked about, where we used aged mice of 57 to 72 weeks old, and kind of followed the same process using spatial transcriptomics as well. And now this then gave us a good kind of platform to compare the results that I saw. And so with my age spatial transcriptomics results, as you can see, I could find very similar cortical regions identified in my young spatial transcriptomics brain samples. And as well, this is kind of the similar graph that you saw that I displayed, where it's plotting the different number of differentially expressed genes following iTBS and cTBS in these aged mice brains. And so Across the cortical layers, iTBS had a greater effect of gene expression in the aged mice, just by slightly. And similarly, for many subcortical regions, there was a large number of gene expression changes as well, which was similar to what I found in my young adult cohort. And so looking at this is kind of a plot of the number of differentially expressed genes that I found following iTBS and cTBS within each of these subcortical brain regions. And because there were a smaller number of differentially expressed genes, I was able to kind of go through them again and look at the different plasticity and the function of these genes and whether they had any relevance to specific neuroplasticity mechanisms. And so this table kind of displays each of what I've categorized. And as you can see, predominantly I found changes relating to synaptic plasticity gene mechanisms following iTBS and cTBS, but definitely to a less greater extent compared to my young cohort. And so as well, there was only evidence of myelin changes following cTBS in these white matter tracts in these aged mice. And so, as you can see already, there's a lot less diverse plasticity mechanisms occurring in this aged mouse brain compared to my young cohort. And so again, looking at kind of these cortical region specific effects, I did the same thing by looking at the motor cortex and performing subclustering of each of my cortical layers. And this is a similar table, but what I wanted to show was that indeed within the cortex itself, iTBS and cTBS predominantly does alter synaptic plasticity mechanisms, and a lot of these were downregulated following both of these stimulation protocols. And similar in the somatosensory cortex, as you can see, the effects of these stimulation were more specific to synaptic plasticity mechanisms, with no evidence of kind of myelin or little evidence of intrinsic plasticity mechanisms following iTBS and cTBS in aged mice. And so kind of from these results, not only from using spatial transcriptomics, I have something to compare. In the young adult brain, we saw changes in kind of synaptic intrinsic plasticity mechanisms and possibly specifically myelin plasticity targeted by iTBS in the motor cortex in the young adult brain. But this kind of shifts as the brain ages. And so there's a lot less diverse plasticity mechanisms occurring. And so not only have I shown through spatial transcriptomics that the effects of stimulation are brain region and cortical region specific, they're also specific to different protocols, and age has an effect on how these plasticity mechanisms manifest as well. And so one of the things that we've shown from this is the need to kind of consider the effect of aging when optimizing rTMS protocols for different populations, and whether a one-size-fits-all use of rTMS in young adults and aged cohorts could be inducing different plasticity mechanisms at a molecular and cellular level as well. And so seeing whether this kind of occurs at a functional level would be something that would be interesting to follow up with from kind of these studies that I've done. And so that was kind of a lot of my preclinical mouse work, looking at the effects of trying to use transcriptomics to map the gene expression effects of stimulation. And one thing that I wanted to try to implement in my PhD was to see how this actually translates to a human brain. And so from our lab, we've produced a lot of preclinical rodent research, but since then, there's no actual direct characterization of whether these kind of synaptic intrinsic or myelin plasticity mechanisms are actually occurring in the human brain. And so in order to do this at the cellular level requires obviously invasive procedures. But one thing that's kind of been growing within the field of neuroscience is to be able to use what we call neurosurgically resected tissues from the human brain to study kind of these effects. And so in some neurosurgical procedures, the removal of tumor tissue deep within the brain requires access to, and with that requires removal of kind of healthy cortical tissue that is actually often discarded in the theater. And so, as part of my PhD project, I wrote ethics in collaboration with the Sir Charles Gardner Hospital, which is the local hospital here in Perth, to collect this cortical tissue and then take it back to our lab and actually study the effects of rTMS directly within the human brain. And so this is unpublished work that I'm hoping to kind of publish within the next few months, along with my supervisor, Alex. But as you can see here, we took this to the lab and kind of applied stimulation using one of the what is a standard clinically used figure of 8 TMS coil. And we were interested at looking at iTBS and 10 Hertz, which is commonly used in the clinics as well with rTMS. And so a big part of my project was to look and perform, again, spatial transcriptomics on these human cortical slices to see the effects of stimulation on gene expressions within the specific cortical layers. And so this is kind of an overview of the different samples that we currently have. It's not a lot, but we kind of sample on an opportunistic basis, just based on the cases that we could get. And so we had a range of ages and from a range of brain regions as well, which again does have its caveat that it does come from different brain regions. And so how the comparability of each might be quite hard. And so this is kind of an H&E example of the human brain slice that I had for spatial transcriptomics. And as you can see, just using unsupervised clustering to see what spots had similar gene expression profiles, there is evidence of some specific cortical layering as well. And so one thing that I forgot to mention was that from each patient, we got kind of a 1 centimeter cubed piece of cortical tissue that we sectioned, and from each patient performed iTBS and 10 Hertz and sham simulations for both from the same patient. So we did this for each of the 4 patients so that we could have a within patient control as well of this effects of stimulation. And so because I was interested in seeing in the human cortex what specific effects are on different cortical layers, one thing that I did was to use kind of histology to guide my manual annotation of specific cortical layers. And then from that, perform differential expression analysis, comparing iTBS with its own patient's sham simulated sample and 10 Hertz with the same patient's simulated sample as well. And so unfortunately, one thing that you can see from this graph that plots like the number of spots of each cortical layer is that there is great variability in the kind of human cortical slices that I got. And so not all of them were of great quality and displayed really nice cortical layering. And so I only chose to perform differential expression analysis on rTMS stimulated cortical layers that had a good within patient sham control. And so with my next few slides, when I display the overlaps of differential expressed genes, you can see why there's a mismatch of different donors and stuff like that. And so kind of what briefly I want to show is that we looked at the differentially expressed genes within each patient and then looked at the overlap, which is bolded in the middle across each of the donors. And so this then gave me a good idea of the genes altered by each stimulation protocol, so iTBS and 10 Hertz, that were common across all the individual donors that I looked at. And so from kind of these genes that were altered across multiple different patients, what I did was, again, went through and characterized each of the specific functions of each of these genes into different neuroplasticity or neuronal processes. And as you can see, there is evidence of multiple plasticity mechanisms occurring across all of the cortical layers by iTBS and 10 hertz, though it does vary between cortical layers. And so from this, one thing that we also wanted to do was to look and perform actual patch clamp electrophysiology on these neurons within these cortical slices. And so this was done in as a supplement to a lot of the spatial transcriptomic work. And so we made sure to have enough brain tissue for those before we did any extra patch clamping. And so we could only do it following iTBS. And so this is kind of just what it looks like under the microscope. And we could observe several healthy neurons after several hours of the brain being removed. And so these are some representative traces of a single action potential and some action potential firing activity within layer 2-3 neurons that we saw in our slices. And so from that, we were able to assess the intrinsic neuronal properties of several neurons. But as you can see from some of my sample sizes, due to the limited number of tissue that we have, there is a limited number of sample size of each kind of iTBS and iTBS sham stimulated cells. But one thing that we were, I guess, excited to see was that there are some differences in things like input resistance and max final frequency, where following iTBS that actually increase, showing that there's an increase in neuronal excitability following iTBS. And so from kind of these studies, it is exciting to see that there is a reflection of iTBS inducing similar neuroplasticity mechanisms that we see in the rodent brain as well. And so just to kind of wrap up all of the work that I've presented, through a lot of my spatial transcriptomics results, we can see that rTMS effects are dependent on a number of different variables. As well, in supplement to a lot of my spatial transcriptomics works in mice, I've provided the first kind of direct characterization of the plasticity mechanisms induced by rTMS in the human cortex. And indeed, we can see that rTMS is able to act on multiple different neuroplasticity mechanisms simultaneously, which you can't do if you're looking at things in isolation. And so while larger sample sizes are required to kind of perform a more in-depth analysis on a lot of my rTMS effects that I've seen in my human brain slices, it is exciting to know that a lot of the kind of plasticity mechanisms we're seeing in the rodent brain does actually occur in the human brain as well. And so I would just like to thank my supervisors and acknowledge Dr. Alex Tang, who kind of guided me through a lot of this work. And yeah, I'm happy to take any questions regarding any of the work that I've presented as well.
Thank you so much. That was an amazing talk. Do we have any questions from the audience? I'm just going to show the gallery mode to see where people are at. Nigel has a question.
Thanks, Leanne. Hi, Rebecca. That was a really fascinating talk. Thank you very much. I was quite interested to see the changes in the inflammatory markers in your last study there. Do you mind just expanding on that a little bit? Were they increasing or decreasing? And if you know off the top of your head, what kind of inflammatory markers were changing?
Is it in kind of this human?
This one, yeah.
Yeah, so I can't off the top of my head remember the specific inflammatory markers, but there were evidence of some things like I think interleukins were kind of increased. But yeah, I think because there's so much data, it's hard for me to recall the specific up and down regulation of genes. But one thing to note with a lot of my human data is that within just a single gene, let's say interleukin B or something, across different patients, the regulation of that gene could differ as well. And so one thing that a lot of my human kind of results showed was that there was a lot of inter-individual differences as well. And so whether that meant that a patient maybe started with higher levels of that gene and iTBS kind of reduced it, or whether one person started with lower and increased, kind of is hard to really distinguish. But yeah, that was one thing to note in a lot of my human results.
Right, thank you.
Okay, if I ask a question?
Go
Yeah.
ahead, James.
Cool. Yeah, I really enjoyed it. It was great work. So I've just posted it as well, but most in your mice work, most of the effects are for cTBS,
Mhm.
from what I could tell. Which I started kind of trying to think what could be a reason for that. And so I was just wanting some clarification from you on what the mice were doing when they got the rTMS. So were they active, awake? Were they moving? Were they asleep? Were they sedated? Because we know from human studies that the level of activation, so we say, in the motor system, if you voluntarily activate either during or immediately after a theta burst protocol, it can very much change the nature of the effect.
Mhm.
And iTBS is a longer protocol to deliver than cTBS, even though they're both relatively short compared to other types of rTMS. So I'm just fascinated to understand a bit more about the setup and how you go about it.
Yeah, so the way that I delivered the protocol for mice was that I allowed them to be freely moving. Um, and so that explains kind of why I had a 7-day habituation period to get them kind of used to the feeling of having the coil on the head. So I'll kind of just have them on my arm or on the table, and I'll just follow them with the coil. And so trying to keep it within the same position to target kind of that sensory motor area within the mouse
Experience
head.
a startle effect from the TMS pulse sound?
No, so we habituated them to that as well. So we would play the machine kind of in the background during that 7-day process. And with kind of these rodent-specific coils, they don't have the same clicking sound that you hear with kind of the human rTMS machines as well. And so that sound isn't something that they hear. They just kind of hear a machine by like whirring in the background.
Thank you.
That's okay.
Maybe I can go next. Hello, Rebecca. Thank you for your talk. Just wanted to ask you about your human data, so the human sample. And I think it's great that you've tried to translate your results to a human sample, but I'm just wondering how close you think that is to the actual situation in a human brain where it's an intact system versus you're taking a sample out of someone's head and then placing it in a dish, and in relation to the stimulation used as well. So in the intensity of the stimulation, the anatomy of the situation, how close is your human sample to the actual real TMS situation in an intact human brain?
Yeah, that is a very good point that you've brought up. And yeah, while it doesn't reflect exactly what's going on, we've tried to keep kind of these brain slices alive, so they are electrically active. And so whilst they aren't the exact reflection of what could be occurring, especially since you lose also the skull, that might influence some of the intensity of the electromagnetic field. It is, I guess, a step closer to having a lot of what we see in a mouse brain observed in the human brain. And so whether, yeah, things like seeing gene expression changes in the human cortex gives us a kind of a good reference that I think a lot of myelin changes have been observed following stimulation protocols in the rodent brain and slowly being translated to human kind of studies. And so having this as kind of a reference point to see, okay, there are actually similar changes occurring at a cellular level in the human brain, I guess is something that this work is really kind of showcasing. But yeah, it is a good kind of thing in the back of my mind that it doesn't actually fully reflect what is happening in a clinical setting.
Thanks.
Could I ask something?
Go ahead, Andres. Yep.
Oh, thanks. Yeah, great talk. Congrats on the submission and the
Thank
papers.
you.
It's well done. So people think about, or have thought, should I say, about part of the mechanism of these neuroplasticity TMS protocols as, involving depolarization, triggering action potentials. So you've described changes here without that occurring. Can I ask if you can offer any thoughts on actually how those changes might come about, what a field might be influencing?
Yeah, so with kind of these sub-threshold stimulation protocols, one thing that it could be influencing is things like more intrinsic plasticity processes. It is still inducing kind of depolarizations within these cells. And so the repetitive delivery of these kind of pulses might be inducing things that don't actually require action potential firing. So kind of shifting the threshold of cells and things like that. So I think that's what is occurring at the cellular level in these mice, I guess.
Oh, yeah, so like more subtle threshold shifts and
Yeah,
that sort of thing?
yeah. So yeah, the stimulation is maybe not producing actual potential firing, but is inducing kind of allowing these cells to kind of still depolarize as well.
Thank you.
Go ahead and ask a question. Actually, I was curious to find out whether you had or the lab has plans to look at other clinical mouse models, expanding that work within clinical mouse models like Parkinson's disease.
I am not too sure what whether we're moving towards using different mouse models. I think some of our lab does use it on kind of Efrin mice, which have kind of abnormal visual circuitry. But beyond that, I don't think we've moved into using different models yet. So yeah, I can't really comment on whether that is, I guess, something that we're interested in going through. But I guess within the realm of aging, things like Alzheimer's and stuff might be an interesting kind of follow-up to see what rTMS is doing in kind of those more disordered brains. But yeah, a lot of our work has focused on kind of the healthy, normal brain.
I'm just checking if folks have any, feel free to, because yeah, we have 23 of us here, feel free to chip in. And I know the whole lab group is in Jamie's Zoom account. So yeah, feel free to chip in and tell us more about any new developments you guys might have with this type of work. No worries. Yeah. Do we have any further questions? I guess whilst we have, I think we have another chat. Yep. So whilst we have kind of 23 of us here, I'll do another plug. Well, before we do that, I'd like to thank Rebecca again for presenting this really excellent work to us today. Thank you so much. And I'll do another shameless plug for asking your lab members to present new work so that it gets the kind of attention that work deserves. We have a lot of us here from all different parts of Australia. Because the talks are presented and recorded on YouTube as well, this can be like a nice little forum for you to showcase the international reach of your work. Really handy to, put in CV lines, grant applications. Feel free to push your mentees to present to us in this nice platform. So I'm just going to check. I don't think we have any further questions. But yes, this will be uploaded on YouTube in a couple of hours. I don't think we have further questions. Any further questions? No. Yeah, so if you have a person you think should be showcased, if you want me to give a little nudge, email me and I can go ahead and invite that person to go to present their work. We are up to the hour now, but yeah, thank you so much, Rebecca, again for presenting this work. We really appreciate it, and we hope you enjoy the little bit of a break that you have before your Viva. And we look forward to seeing what comes next for you.
Thank you.
Thank you so much. Thank you. All right, bye now.
Okay, see you later.
Thank you. Thanks.